So next, we're going to have a talk by Nick Sauter. I think is joined online by his colleague David Mittan-Moreau. And they are computational scientists from Berkeley Lab, and they're going to talk to us about some of the science and what the science use cases look like.
Okay, thank you. So thank you for the introduction, and thank you for the two talks today so far. It's just really fabulous, and it almost does my talk for me, I think. So let's see if I can switch to present. So I'm from the biosciences area at Lawrence Berkeley Labs, and so is my colleague David. David, are you online? You might
I am online.
Might want you for answering questions later. So I'd like to share some of sort of our perspectives as frequent users of the LCLS facility for structural biology, and what our expectations are for the next few years, starting from where we are right now. So I thought, since we have a very diverse computational group, I should define my terms. So what is serial crystallography? And actually, it's different from traditional crystallography, where we have a protein crystal, one of them, we rotate it in an X-ray, in a hard X-ray beam, and collect a diffraction pattern. The rotation allows us to do the whole pattern. And the concern here is always radiation damage, both general radiation damage that decays the whole diffraction pattern, and specific chemical damage to things like metals. So serial crystallography is a way of getting away from that. Instead of taking a crystal and rotating it over a time period of a few seconds, we collect thousands of diffraction patterns. In this cartoon, we're sending a stream of crystals through an interaction point where we hit it with X-ray pulses. So each crystal experiences the first shot, and we never accrue that radiation damage. So we get much better results that way. So it's the advantage of that is in the traditional approach, we're always trying to protect from radiation damage by going down to liquid nitrogen temperatures. But here, with serial crystallography, collect everything at room temperature. And I'll say more about that in a bit. And then, because of the way we're doing it, as I said, we're using short pulses, especially at LCLS, where it's a femtosecond pulse. We're actually free from radiation damage. But don't take my word for it. We actually do the control experiment every time. So in the forward direction, we're collecting diffraction pattern. And at right angles, we have a spectrometer here where, as I said, the metal atoms are often where we see specific damage. But here, you have a spectrometer that collects X-ray emission from metal atoms, such as, iron, manganese, cobalt, nickel, and so forth, that are in our metalloenzymes. So we can make sure that we're observing the metals in the right oxidation state. The other thing we can do, because we have, especially at LCLS, the femtosecond time structure of the pulse, we can do pump probe experiments, and we can actually watch enzymes. We can synchronize with a trigger point of some sort, and we can synchronize enzyme reactions and look at time evolution. So, for example, with in photosynthesis for photosystem 1, we're capturing sunlight and converting it to chemical energy, and we can actually follow this on the picosecond to nanosecond time scale. Also for photosystem 2, that's where we're generating the oxygen that we breathe, and it's governed by a little cofactor in the enzyme that has manganese and oxygen atoms. And here we actually have time points that are triggered by flashing with a series of 3 or 4 different green laser flashes. And we can actually see an oxygen or a water molecule come in as a substrate. So that's more or less on the microsecond time scale. And then sort of at the millisecond to second time scale, there's plenty of biological reactions. For example, here, the production of penicillin, where the last enzyme step is an oxygen atom coming in. And we can we can synchronize that by actually incubating the crystals with oxygen, or we can mix a compound. We can mix substrate into a crystal using this. This is the Berkeley Lab drop on tape system of sample delivery. So we actually deposit droplets containing crystals here, and then that belt moves into the interaction point with the X-rays. Oh, and I'm actually showing it live here. So these large droplets are 2 nanoliter droplets containing crystals, and then these little tiny droplets that are being deposited on top of it contain substrate. So we can actually, with millisecond time resolution, follow an enzyme reaction. And so, okay, so now let me focus in on the speed at which this is done. With current instrumentation, we're doing this experiment at 30 droplets per second, 30 hertz. Why is that important to me? Well, it's actually really difficult to do one of these serial crystallography experiments. Like I said, in the traditional approach where we're rotating, we collect the full diffraction pattern. In a serial experiment, we're only getting a slice of the Bragg spots for any given shot. So we have to merge the data from tens of thousands of crystals. And all the time we are worrying, do they have isomorphous unit cells, or do they all diffract out to high angle, which gives us the largest spatial resolution of atoms in the protein? And so if the crystals aren't uniformly good and of the same unit cell, we can't merge the data together. Also, every Bragg reflection that we collect in a serial shot, in the serial crystallography shot, has to be corrected in intensity by an unknown scale factor, which introduces all kinds of computational complexity, and we're not even agreed on what the right algorithm is in the field to correct these intensities. These intensities are important because from these Bragg spots, we get what are called structure factors that are the Fourier coefficients for calculating the electron density map for building in the atoms. So all these measurements are in a computational pathway to get to the final answer. As a result, in contrast to rotation crystallography, we have to collect tens of thousands of shots. So, and all the while, we're going to be continually assessing the data quality to decide what's the stopping point. So this is really what's happening in the control room at one of these experiments. So one of the PIs in our division, Jan, says, for a 5-shift photosynthesis experiment, we brought 15 milliliters of sample. It took 22 weeks of a full-time employee to create that protein. That's what's at stake here. So everybody in the control room is in a state of panic. Okay, here's what everyone is doing. This is actually the Beamline scientist. He's an employee here, and he's running the DAC, the data acquisition software. He's dealing with issues like, is the beam getting into the hutch at all? Here's a postdoc who's looking at sample delivery. Is the beam actually hitting the sample? Or do we further optimize it? This is another postdoc who's looking at the X-ray emission spectroscopy, asking if we're in the right oxidation state. And this is my team. I didn't even put it here, but we have software, CCTBX or dials, for interpretation of the diffraction pattern. So we're looking at the resolution limit. Are the crystals isomorphous? And if we merge the data, do we have a complete data set yet? Can we stop data collection? Then there's another postdoc who's taking the Fourier coefficients and asking, do the results support our hypothesis? And all of this feeds back into the PI, asking, what is the sample or time point that we should collect next? So considering the enormous amount of effort that's going into this, the most important mitigation that I can provide as a sort of semi-computer scientist is immediate data reduction with a 10-minute turnaround time, evaluating the final science in order to know if the experiment is on track. So I don't want to give the impression that this is a goal we are striving for. We have achieved this goal, and we've been doing this for the past 8 years. So we have a long history of collaborating with LCLS, ESNet, and NERSC on the assessment pipeline. It started all the way back in the Edison days in 2014, and it's continuing through now for 2027 with Drodna coming online. So and it's been a continual evolution. The beams have changed, the detectors have become better, the compute clusters expand and the algorithms change, but still, and here's where we are currently. We have a Jungfrau 16 megapixel that is clocked at 120 hertz, and that in 12 hours produces 150 terabytes of data. You know, we're not going to be talking about megahertz in my talk. Protein crystallography, the most that I can possibly envision right now is 2 kilohertz. So let's talk about that for the next few minutes. So here's what we have now. We have a dual implementation. We can either do the calculations here at SLAC at S3DF, or we can ship the data over to NERSC like we've been talking about. We have a petabyte disk allocation there. At NERSC, we would use 32 CPU nodes in the real-time queue on demand for our 12-hour shift. And all of this is supported in the data analysis by a MySQL backend. And then we pipe the data back to the users. Okay, so that's where we are now. So where do we want to be if we're going to do serial crystallography at 2 kilohertz? So that's the rest of the talk. So the first thing I'd like to know is I want a clearer understanding of the goals. So I'm going to ask the questions. I don't necessarily have the answers today. What algorithms do I want to use? You know, how fast does the feedback have to be to the experiment? Can we fail over to another facility if the one we want to use isn't available? And so I'm going to be asking questions about data portability or data processing portability and standardization and automation. So that's my talk today. So what can we accomplish at 2 kilohertz? Anybody want to contribute to that? Okay, well, here's some ideas. So some of my colleagues have discovered we can do crystallography on the small molecules in material science, chemical crystallography. And there's been a pilot program in place. So in 24 hours, recently, we've been able to solve 11 small molecule structures. So let's move that up to 2 kilohertz. So now we're talking about 1,000 structures solved. So it's a little bit mind-boggling. That's not going to be one group mailing in samples. That could be hundreds of groups mailing in samples. So clearly we're going to have a much increased capacity. So now let's go back to the protein science side. In recent years, this is a one beam time we had. We had 48 hours of data collection. We looked at 12 different types of enzymes. We collected 54 data sets. So for each enzyme, clearly we sampled a few different conditions. Now this will be able to greatly expand this. So think about what serial crystallography can do. Since we're studying enzymes under the real conditions of chemical reaction, we can study the factors that go into it. We can study temperature dependence, time dependence, pH, oxidation state, and even perturbing the protein with electric field and so forth in order to get a good understanding of the conformational flexibility of a protein that would lead to its biological function. So there's a lot of science we can think about if we can just do more data collection. Drug design. Ever since COVID, I've been seeing several papers every month about drug screening. And the nice thing about shifting from traditional to serial crystallography is that at cryogenic temperature, things are distorted. So this is a drug candidate binding to an enzyme, but you see it's different at room temperature and cryo. So room temperature would reflect more of the conformation of the real active enzyme under upper endo conditions. So I think it's to our advantage to move everything over to room temperature. So that's the pitch. And then furthermore, what would a pharma company do if they were they know this is a protein that's a drug target. How do we develop drugs that might bind to it, that might be enzyme inhibitors, or that might modulate the function of an enzyme? So what they would do is they would take the structure of the protein, which is already known, and do a computational screen with hundreds of thousands of possible drug fragments that might bind. So they're going to narrow the field down. Then they would use fast screening to determine what's binding to the protein. And in the past few years, we've seen more and more papers saying that my primary experimental screen is going to be crystallography. So we're going to just simply do thousands of independent crystal structures, with each of which has a chemical fragment bound to it. Okay. And papers like this, but I don't think I've seen any papers using an XFEL, an X-ray free electron laser, but I think that's coming soon. And clearly there'll be an advantage at 2 kHz. But there are other modalities. This is not crystallography anymore, but it's small angle scattering of protein solutions. And under certain circumstances, you can get structural information, both shape and size, just not to atomic resolution, but pretty close. X-ray absorption spectroscopy is another one. If we really want to study those metalloenzymes and what's going on at the metal, we can do X-ray absorption spectroscopy, but it requires a lot of data collection and it's very noisy. So if we can simply increase, the signal to noise by increasing the number of shots, that's to our advantage. So I don't have the answers. Some things will benefit and some things won't from kilohertz. So the spectrum is going to go all from the negative side on the left all the way to the favorable side on the right. Some of the experiments will probably stay at 120 hertz. And I think I might include the photosystem as one of those, that we really require the way our tape drive conveyor belt sample delivery is set up now. We're stimulating the crystals, as I said, with 4 laser beams, optical laser beams, before the sample gets to the interaction point. It's very hard for me to imagine doing that at 2 kHz. We're sort of constrained by the sample delivery method and also by sample availability. So I would say in these types of cases, maybe we're better to look for bigger crystals. So right now we're using quite big crystals, 20 microns to do these studies. On the other hand, the chemical crystallography that I mentioned before, here, you don't do serial crystallography at all if you have a single beautiful crystal. What you do is you maybe synthesize something and it's a powder. You can't get big crystals, but you want to know the structure. So you use serial crystallography. That's the pitch for using XFELs for chemical crystallography. And there are some proteins that are like that too. And certainly I think I consider the drug screening and the drug repurposing as good candidates for kilohertz. Okay, so that's what our goals are. Now, as I said before, we have a sort of a computational problem in serial crystallography where we have to do sort of data manipulation or data scaling of each of the Bragg spots individually. Also, what are the goals? I mean, in serial crystallography, we already know the structure of the enzyme. We're looking for details. We're looking for a drug fragment. What would happen if you added a methyl group? Or what if you made a single amino acid mutation? How would the structure change? So we're looking for tiny little changes. And as a consequence, the data have to be accurate. So we're always striving to do better in the analysis of these Bragg spots and how we scale them together. So about 10 years ago, we came up with the idea, we developed software called nanobragg or diffract, where we're going to use diffraction physics to model the data. So here's the experiment. We're going to model these pixels, 1 pixel at a time, 1 photon at a time, taking into account things we know about the experiment, such as the beam isn't exactly monochromatic. So it's a little bit more difficult to analyze the diffraction. And also, as I said, there's these different scale factors that have to do with crystal imperfection, crystal mosaic structure. So we came up with this is actually a textbook equation from, I think, the 1910s. So the idea is using physics to model the data with a parametric model, and then changing the parameters of the model in order to best agree with the data. So the promise is it's going to give us great results for tiny biological differences. It works great for simulated data. And the challenges are that maybe it's an older approach. We did use GPUs, but it's in CUDA, so it's a little bit hard to maintain. It probably requires about 64 GPU nodes for a reasonable calculation. And the terrible thing is it works great for simulated data, but not so good for experiment. And why is that? Well, clearly there are things, the systematic effects on the data that defeat our goals. So things like beam polarization here, where the diffraction is more intense in this quadrant than in this quadrant, and things like this, where the beam is hitting, or the diffracted ray is hitting the detector at an angle. These are things we might be able to correct with physics. But then there's other effects, such as absorption and shadows that are cast on the detector, and also the aspects of the mosaic structure of the crystal. Crystal isn't perfect. It's got these little mosaic blocks that are misaligned with respect to each other. It's really hard to have a physics model that models these effects. So we're going to hear a talk, I think, from Kevin Dalton today. He has the approach of using variational inference to scale the Bragg spots. So essentially training with his program Careless, the neural network to learn how to scale the data together to correct for systematic effects with no physics. So it's great. It's very promising. It's not scalable in memory. It only uses one GPU node. I see. And it relies, therefore, on stochastic training, sequentially putting the data through the analysis to loop through all of the images that we collect. And the other challenge is it's not been integrated into a 2 kHz modality. So that's for us to do, and I'm excited to get to work on it. Okay, so is the experimental feed fast enough? Well, of course it isn't. Jan says we know what Jan said already. It takes 22 weeks to prepare our protein. Aaron Brewster says, oh my word, that's 42 years of sample prep for 5 shifts at 2 kHz. Okay, so in my view, this whole rule that we had about providing feedback in 10 minutes, that goes out the window. Because in those 10 minutes, we're going to burn through 40 days of work preparing sample. We have to get down to 1 second turnaround. So that's what David is working on there in the Zoom background. Why did it come up in the first place that we were going to have 10 minutes? Well, the way the DAC works, data acquisition, is that we divide data acquisition into 5-minute runs. And then the data is sort of staged on the fast feedback flash memory, but it's put into a file system. Only then do we take the data and pipe it to the analysis clusters, such as NERSC. Actually, it has always been possible to get feedback in 1 second if we put our nice Python code inside the DAC. And we actually did it once. The danger there is that we're going to crash the DAC and we're going to lose all the benefit of doing this. So after that experiment, we never did it again. We just adhered to this model of data acquisition, then piping the data to a cluster, and then analyzing the data to get the feedback. But David is saying, okay, let's just get rid of all this. The goal is to take the data right from the detector, pipe it into the analysis cluster, and immediately get the feedback. And so that's what we hope to do. And actually, that's what he's doing. So we heard actually one approach today. I would say this is the same thing, but here we're using the 0MQ messaging pattern to take data from the detector. And in this case, we're doing these experiments at the ALS Gemini beamline at Berkeley Lab. So we have 4 nodes that are receiving data. They're running MPI. The tasks of the MPI workers the ranks are different. So for each node, there's going to be a broker that receives the data packets, and then that broker allocates work to the workers. When new results are available, they get sent back to the display. And when all the workers are done, the data can or cannot, you can do it or not, you can archive the data to a file system. So that's what he's doing now. And here are the results. So here's, over time, we were doing 5 runs, each about 20 seconds each. And on the y-axis are the different ranks. So there's control ranks, there's broker ranks, and there's archiver ranks. And in the middle, these are the workers that are actually getting the data. And what he's doing is he's running a data analysis pipeline, the traditional one that we have always used. And it goes from, anywhere from 1 second to 20 seconds per X-ray event with a median of 2.5. So what can we do? Yeah, I'm working on it. So what can we do to go from our 2.5 seconds down to just milliseconds? Well, there's a number of tools out there already. So this one is ResoNet. It's from Derek Mendez here at SSRL. And I think you mentioned something that I'm not aware of, Yana. I think there's another program out there somewhere. So what he's doing is he's trained a neural network on GPU to identify Bragg spots. So the green trace here is just counting Bragg spots, and the red trace is discovering whether they're diffracting to high angles and giving the high resolution data. And then this last metric is asking whether the crystal lattice has split, is the crystal damaged, and in that case, we would want to throw out that data. So he says it's easy to train the data with the neural network simulated data. The disadvantage is it hasn't been tested at kHz yet. So we got to do that. And then remember I said that these crystals are going to be bad because the lattice is split in his analysis. So can we convince the user that this is sufficient for event rejection? Because as we know, we're not in the future going to be able to keep all the data. We're going to have to get rid of some of it. So that's kind of where we're headed with this. So now, I didn't actually talk about what's in the data processing pipeline. Spot finding, indexing, which means finding a lattice, and then integrating the photons in those spots and then merging all the data together. So the Toro indexer was developed at the XFEL light source. And here, it's not a neural network, it's a straight GPU algorithm, but it's a new algorithm that performs in fixed time, in contrast to those results that David showed. So things like fixed time would be important for assuring that things, keep in lockstep at the millisecond timescale. So I think this is very encouraging. It's using PyTorch, so we don't have to worry about CUDA at that direct level anymore. The challenge is, again, in order to benefit from parallelism, you have to pack the data differently. So you have to pack 1,000 images together, send it to the GPU, and then work on it there. It hasn't been tried at kHz, and so there's a lot of work to do there. And you know what? I'm aware that the other sort of our competitors at DAISY developing the XFEL software package, they've been thinking along the same lines here. And they've taken their software package and taken the CPU code, but just optimized it. So instead of sort of 2 seconds, they're now taking 500 milliseconds to analyze as latency. So I think there's going to be in a year's time or two, there's going to be a lot of options here. So then we asked one question too many. Can we actually send our data to NERSC from the ALS? And we know at 2 kHz, we're going to need 820 gigabits per second to get the data there. And according to Google, ESNet offers 800 gigabits per second. So we're going to be taking over all of ESNet to send our data to NERSC. And then we asked, can we actually run at this rate? And over the weekend, we could not. So there must be a network engineer somewhere that can tell us how to get to the fast network, because I don't know how to do it. Okay, so I'm probably over time. I'll make things fast. Is data processing portable? Well, actually, no. The graduate student that's keeping track of the data is running as a function of run number. They're typing in words that indicate what the experiment was, but there's no standardization. there's no understanding this if you're outside the group. Then there was another grad student running the analysis. He was typing in these words manually for each run, and then putting that into the MySQL database. Now, what if the experiment crashes or the nurse suddenly crashes and you want to go to Oak Ridge? So you want to bring your MySQL database. No, it's not portable. All this has to be done. And this is manageable at 30 hertz or 120, but not at 2 kilohertz. The file paths that we keep track of are specific to a particular facility. So if we want to bring this data to Oak Ridge, this file path is no good. So shouldn't we be using a distributed file system? Somebody might be able to help us with this today. So I'm looking forward to the conversation. So I think I'm out of time. So I will relinquish. Oh, well, I just put a conclusion here. So these, just in summary, what I've said so far, these are the things we would like. I don't really think I have answers to the questions, just a lot of questions. But clearly this is the direction we're headed. But I have one comment. We don't need all of these things at once. Even just one thing that I mentioned today, like the ability to send, to know what our speed is sending data over the network. Any one of these things can help us today. So I think this is going to be a process where everything is going to benefit us. And I really like to have, I like this opportunity to have this conversation. So here are the people. Now, of course, I have a lot of more collaborators than this, but these are only the people who contributed to the actual work I showed here, and we're working together now. So there's a lot of other people, and I apologize for not acknowledging you. So thank you.
Got time for questions?
Oh, nice to see you.
Nice to see you too. So I think I understood the motivation for the 1 second feedback was that, to amortize the investment in creating the samples. That's what the rate at which you would need the feedback.
The last slide, I don't know if I can answer some of the questions. This kind of shows that we envision self-driving labs. So that's some of the purpose of the 1 second
Okay,
feedback.
because I was gonna say at 1 second, you're not having to be making human decisions anymore. So that whatever's consuming this feedback is doing it automatically.
Yeah, it's going to be a self-driving. So these different arrows are the different types of feedback. So some of them are internal to the tape drive. Some of them are based on the diffraction pattern. And then some of them, you compute the electron density all the way downstream. And then you can still give some kind of feedback through the way you set things up. Yeah. Yeah, there's a lot of exciting stuff that we haven't thought about or done. Okay.
So it sounds like then that there is clear automated actions to take based on the feedback. I mean, like the control loop is.
Well, just to adjust the tape drive so that it's actually in the beam, things like that. Because if there's no diffraction, that must mean that you're not hitting anything. And so then you can re-steer. So there's literally a graduate student doing this. I actually think he's a postdoc in our group. Now,
Requires a PhD to.
yeah, we're actually doing this manually, but this is why David is doing this work so that it becomes a self-driving.
So are these more just like control decisions, or is there any actual like science decisions being made autonomously?
Well, the science decision that I described was the one where, do you have enough data to complete the Fourier transform and move on to the next sample or chemical condition? So that's what we're doing now, but it's a very manual process. Whereas, so if I were a drug company screen, I would have a large device like this, a fixed target where I have protein crystals all over, but I would have little squares where each square represents the addition of a different chemical fragment. So that's easy, right? If we can determine using this type of processing that I have a complete data set and that it answered a scientific question, then I could use my little chip to move over to the next sample. So absolutely, all of these things lead into.
I mean, the only thing I would add is that's not very far from actually automating the scientific search, right? Like in materials, you cover a wafer with a chemical variation across the wafer. And now, not only can you decide that you've got enough to reconstruct the crystal or the chemical, but it can tell you where to move the wafer for the next interesting point, for instance. And that way you could do guided search.
More questions?
All right, thank